Hello judges, we would like to present our project, High Efficiency Multi-Chambered Microbial Fuel Cells and Applications. Theoretical background. The microbial fuel cell, the MFC, is a bioelectrochemical technology using microorganisms as a catalyst to convert organic waste into electricity. An MFC compresses two electrons, an anode and a cathode, and relies on the capacity of certain microorganisms in the substrate to use the oxygens in the cathode as their N-terminal electron acceptor in their anaerobic respiration. When microorganisms decompose the organic matter in the substrate, they simultaneously release protons and electrons, and the protons infiltrate to the cathode through proton exchange membrane due to the ion concentration gradient, while the electrons go to the cathode via the wire and generate electricity. Finally, the electrons will combine with the protons and the oxygens in the cathode into pure water. In our research, we improved the generation efficiency of the MFC through microorganisms and the MFC device in order to apply the MFC device in the toilet to make a self-power supply toilet more realizable. To find appropriate strain, we selected them from urine. After purifying the microorganisms in urine, we put them in the MFC and measure the voltage. And then we identified the superior strain, which can perform higher voltage in urine-based MFC in order to find their scientific names, biosafety levels, and characteristics in urine. The improvements of the MFC. To increase the power and current, we set up two traditional MFCs in parallel and connected two cathodes into one chamber as a three-chambered MFC. And protons infiltrate to the cathode from two anodes, which can increase its ion concentration gradient. Therefore, we made a hypothesis that three-chambered MFC can generate higher power than two traditional MFCs in parallel.
The results. As shown in figure 3, we can see that strand A and strand B had significantly higher voltage than others. Therefore, we identified them. And since strand B originally habitated in urine, we used strand B as the microorganisms in our following experiment. And the three-chamber MFCs. The current of three-chamber MFC was about, after 20 hours, was about 0.3 milliamp, with a current density of about 60 milliamp per square meter. And the average open circuit voltage was about 0.52 volts. As for the power of three-chamber MFC was about 4.38 milliwatts, with a power density of about 1.65 watt per square meter. As shown in figure 7, three-chamber NFC can generate about 4 times higher than traditional NFCs, which proves our hypothesis. And since the power density of three-chamber NFC was about 4 times higher than non-traditional NFCs, we inferred that the 5-chamber NFC could reach about 16 times higher than that of traditional NFCs. So we conducted an experiment. As shown in figure 9, the power of 5-chamber NFC was about 22.66 milliwatts, with a power density of about 4.45 watts per square meter. And as shown in figure 10, we can see that the power density of 5-chamber NFC was about 2.5 times higher than that of 3-chamber NFCs, and 11.5 times higher than that of traditional NFCs. And the reason why it couldn't reach about 16 times higher than that of traditional NFC might be related to the saturation of proton. And this is our urine generation device. We came up with a device that can generate electricity in the urinal. And first, we set up a container on the bottom of the urinal and connect it with an MC. And then we use administration set to control the rate of urine inflow and outflow. And after that, we use immobilization technology in order to prevent the microorganisms from losing while the urine flows out. And this generation device can not only generate electricity, but also reduce water demand in the toilet.